Right.
I think we need to bring in the spookala bookala.
Spookala bookala. Yeah, spooky, scary skeletons. Okay. Oh, it's me first. Okay, here we go. Welcome to Absolute Genius, a new podcast series from Thermo Fisher Scientific. I'm Cassie McCreary.
And I'm Jordan Ruggieri, and today we're joined on the show by Dr. Nikhil Ram Mohan.
Nikhil earned his PhD in genetics and genomics at the University of Connecticut in 2016. He's currently a staff scientist at the Stanford University School of Medicine, where he specializes in bioinformatics. He's working on some extremely interesting research around SARS-CoV-2 co-infections to help study the virus more.
He also has had a fascinating career journey. We hope you enjoy our conversation. Oh, Cassie, I can't believe that viruses and bacteria would just invade my body without permission. That makes me sick.
Here we go. Back to puns.
So I know, you're researching SARS-CoV-2 and bacterial co-infections. Can you tell us a little bit about this? How does that work? What exactly are you looking into?
So when the pandemic hit, us being the research lab in the emergency medicine department, we were tasked with understanding how viral RNA was working in the host system itself and what the presence of viral RNA in the blood meant to outcomes and whatnot. So the first thing we wanted to really see is how the viral RNA in the blood correlated with the viral RNA in the nasopharynx, because that was the point of entry for the SARS-CoV-2 virus itself. And as we went through the study, we started looking into estimating the copy number or the number of transcripts of the viral RNA, both in the nasopharynx as well as in the blood itself. And traditional methods only allowed us to use qPCR. And we heard about this instrument for absolute measurements using digital PCR. We wanted to explore that as well. So what we did was basically from blood and nasopharynx, collect samples, extract RNA, amplify the N1 and N2 regions of the viral genome, and just estimate how much of it was present in the body. And based on what we found in the plasma itself, we then did some modeling to see how well that would be associated with extrapulmonary complications and whatnot. That was one wing of the study. The other study was basically looking at bacterial co-infections or superinfections in people who had COVID, who had visited the ED. And we were trying to see if the viral RNA in blood was very well correlated with this one test we were using that was able to detect both virus and bacteria in the subject's blood itself. And there again, that seemed to work out really well. It was pretty interesting, actually. We saw that, for one, the viral RNA in the blood, or anemia as it's been called now, was actually a really good predictor of severe disease and a host of extrapulmonary complications as well. And there was also a strong correlation between the viral loads that we detected and the presence of viral scores from the test that we were doing in the second study.
So it's more along looking at if there is some correlation on the amount of virus, the amount of bacteria, and how the virus actually impacts the host, right? So research and looking at if you have more virus, does that cause a more negative impact on the host? Or if you have both virus and bacteria, does that cause a more negative impact on the host? Is that a correct assumption?
Without necessarily going into the mechanism of how things are happening itself, we were just it was a prospective study. So based on the information we had, we tried to make these associations that we were able to based on the viral loads and the 30-day mortality or the need for greater oxygen mechanisms of implementation and stuff like that. Yeah.
What did the research show about co-infections and how it impacted the host?
So we were testing a particular methodology of detecting the viral RNA or signatures for viral and bacterial infections based on host response against the COVID patients that we had in our biobank. And what we saw was that co-infection rates are not very high, but it can differ based on the institution you're sampling from, if you will, right? So it can be as low as 3% co-infection or as high as 10% co-infection in the population that you're studying.
And how do you identify which co-infections to research? I mean, were you looking more just broadly to see, or just in general if they had also had a species of bacteria, or did you actually go in mind thinking of, did they catch E. coli, or do they have some type of other organisms? Were you looking to, how did you decide that? How did you look at that?
So it was not pre-planned. A lot of these correlations are done based on the clinical data that we get after the fact, right? So when the subjects go into the emergency room, if a physician suggests that the subject might have signs of a bloodstream infection as well, and they would collect blood from the subject. And then that sample would be inoculated into a blood culture tube, if you will. And then the colonies that are isolated after are then identified for what organism it is. So of all the subjects that we enrolled in the study, a subset had also had these symptoms that were flagged and blood culture analyses done. So we went in and looked at how many of those subjects had these potential co-infections, what those co-infections were, and how those correlated with the viral loads that we were seeing or the viral scores from the other tests we were seeing.
So, Nikhil, it sounds like this was more researching the impact of the virus and the co-infection on a potential host. It's not necessarily diagnostic. It was just examining potential outcomes from these co-infections. Is that correct?
Yes, definitely. So samples are collected while the patients are already there, and the research was performed after the fact, right? So we were trying to make connections for what could have happened based on the data that we had presented.
So, Nikhil, you're in a really interesting spot in terms of the research that you're conducting, and in that the samples that you're looking at and the organism that you're looking at, humans, you know that there is a viral infection. You know that there's something already there. You're almost taking this already known, right? It's a positive infection, and looking to generate more data to research and inform future prospective health outcomes. So it's a really interesting spot in that, most people think, oh, okay, yes, I'm yes, it's there, or no, it's not there. But you're actually looking at this pool of positive data to see if you could extrapolate for future potential, right? For future information, for future ways of looking at a particular type of infection. Is that right? Can you explain kind of that role a little bit?
I can try. So with COVID, at least back then, when you say positive, that just meant that the virus was detected in the nasopharynx, right? Not a lot of people back then were looking at the virus in the blood itself. There were a couple of studies that had started looking at it in the serum and correlating it with certain outcomes, but they were doing only qPCR back then. So we did look at a small pool of subjects who were positive based on the traditional sense of testing for COVID back then. But then we were looking for the viral loads in blood in subjects who were positive by the nasopharyngeal swabs. So that was how we were trying to differentiate ourselves, right? So we were looking at how, if the virus was able to translocate. So let me be clear. So we don't know if the virus was still replicating in the blood, right? We were just looking for evidence of the RNA in plasma itself. So what we were looking to see is that if we were able to find the viral RNA in plasma of the subjects, and whether the viral load itself could correspond to some of the severe complications we were seeing in those subjects as well itself, right? So it's a combination of using prior knowledge about the subject's past medical history, all of the clinical labs that are taken as part of the routine lab work, and adding an additional layer to that in the form of the viral RNA MIA that we were testing in the lab, and trying to see how we would be able to connect the two.
Makes total sense. Did you do any type of modeling with these co-infections, or how were you able to show that these co-infections may have some impact?
So the test that we performed, it uses a suite of host response markers. So that's based on transcriptomic signals from the host itself. And based on the signals that we get, that test actually can talk about a potential bacterial infection, a potential viral infection, and also give us a severity score. We didn't necessarily do any modeling because the N for that particular study was rather small. It was not a very large N. But we saw that when we compared proportions rather than doing modeling, there was some statistical significance in the difference of proportions between those that had co-infections and those that didn't, with respect to the scores that were being generated by the other test.
That makes sense. I want to talk a little bit about that kind of transcriptomics approach. How did you approach assay design for transcriptomics and looking at those markers?
So that was in collaboration with another third party. That was not something that we had done. So this is an offshoot of a different lab from Stanford, where they had studied this for many years at this point and come up with a set of 29 host response markers that describe all three outcome results, if you will. So that we were really testing that platform against this data. That was not something that we did.
Makes sense. So you were using those 29 markers as a measure of potential outcome for the host. And those transcriptomes, is that are they host transcriptomes or are they viral?
They're host response, right? So we send
host response. Okay.
One of the samples that we collected was just the whole blood in tubes called PAX gene tubes, which can protect the RNA in the sample for extended periods of time. RNA is extracted from these tubes, and then you use an N counter to get a measurement of the number of copies of each of those transcripts that we are looking at.
Okay, so it's those 29 markers are indicative of the overall health of the host, and so you're researching if they came in with an infection of SARS-CoV-2 or a co-infection, looking at those 29 markers to kind of gauge general health of the host.
Mm-hmm.
Correct?
Mm-hmm. And also, to be able to detect a bacterial co-infection, it's a lengthy process, right? So you need to inoculate the blood in a blood culture tube for extended periods of time, and then you need to identify what it is. Currently, antibiotic treatments are pretty empirical based on what the best guess for infecting organism is itself, right? So if there isn't even a bacterial infection to begin with, it's just you're seeing the symptoms, but there is no active bacterial infection. You're treating a patient with an antibiotic for no real reason. This is also a way to do a test. In its current form, it still takes time, but they're trying to shorten that to develop a point-of-care test that can tell you if there's a bacterial infection in a specified amount of time.
So that would be really beneficial, right? Because like antibiotic like resistance and everything like that is a big problem. So if we're just, willy-nilly using antibiotics, so to speak, but we have a tool like this, it could be really helpful to maybe potentially reduce that one day.
Definitely. That's the goal, at least, right? So this was our study was also like a proof of concept to show that test can actually detect bacterial co-infections with high accuracy.
Very interesting. We love digital PCR here at Absolute Genius. So talk a little bit about the digital PCR angle of your research. How did you use digital PCR? How was it helpful?
For one, when we were studying the viral RNA-emia with qPCR, we were finding that of the subjects that we had tested, there was a very low positivity rate. But we didn't know if that was because of the lack of sensitivity in the qPCR mechanism itself, or it was actually the case, right? So we started testing out the digital PCR, and we saw that, if I remember the numbers correctly, with qPCR, our positivity rate was about 1.4%. But with digital PCR, we saw that they jumped up to about 24%. And we are getting absolute quantities, right? So there's no need for a standard curve or anything to get the viral load. So it just made more sense for us to go that route for that particular study.
Why do you think, in terms of the sensitivity, in terms of that huge jump from 1% to 24%, how did that impact maybe the precision or the observations for your study?
Yeah. So the challenge was that a lot of the samples that we had really low viral loads to begin with. If we had relied on the 1% from qPCR for us to go forward in that study, we'd have needed a larger N for us to be able to do further analyses, right? But with a digital PCR, that helped us out because the rate limiting step there was that we were only able to sample so many subjects coming in as well. The higher sensitivity with the digital PCR platform really helped us in being able to ascertain the patterns that were actually present, which we would have missed out relying on just the qPCR.
Did you use qPCR in conjunction with digital PCR in any way, or did you use qPCR to help maybe optimize your assays or at least get up the road for some of those assays?
Initially, at least for about 100 or 150 samples, we did both Q and digital PCR simultaneously. What happens is when we get these samples, the clinical microbiology lab is already making sure that those samples are positive or negative. Ours is just a research lab, right? So we need to do the qPCR to confirm to ourselves that it's the same results that we are seeing. So we did do qPCR simultaneously with digital PCR. And that's kind of how we were able to compare the correlations between the RNA-mic and non-RNA-mic and the viral loads and how those were impacted and all of that as well. So the initial study relied a lot on us being able to do both simultaneously. And once we ensured that we were seeing concordant results, then we moved forward with the digital PCR.
So I know you have a very analytical background. How did you approach using digital PCR, and how did that kind of help in regards to your study?
I think for one, just the absolute numbers help, right? It's not unlike that the qPCR data has never been used before because all you have to do is make sure that you're log transforming the data. But with absolute numbers, it's easier to see the minute differences as well. A CT value from 23 to 24 might not seem as big, but you might still see a lot of differences in the absolute quantity itself. So to be able to parse those out, having the absolute quantities from the digital PCR definitely helped out.
Now, you mentioned a few times that you're doing some coding. Are you the one that's actually running the digital PCR? Do you shadow somebody, or do you work in conjunction with somebody? And how important is it for you to understand the what's happening in the lab for you to write good code for the bioinformatics portions of your project?
Let me start with the latter. I think there's a necessary interplay between the wet lab and the dry lab aspects of what we're doing. If I don't understand the experimental setup, my writing lines of code do not make really sense if I don't know what the appropriate controls are and what my comparisons against are either. As for the initial part of the question, during the peak of that study, I had to get my hands dirty and run a lot of the RNA extractions and running the digital PCR myself as well. So there were others doing it in the lab as well, but this was time-sensitive project. So in order to get information out, we just took turns, and I had to actually do it myself as well.
So it sounds like it's good. It's good to have that experience. If you're thinking about going into being a bioinformaticist, it's good to have that blend of both wet and dry lab experience.
Definitely. And we got lucky that the instrument we were using for the digital PCR was a breeze to set up. The reaction mix was the same as just the qPCR mix, and putting it on the plates, they were just pretty straightforward. So I didn't have to learn a whole lot of new things to be able to do it. So that was easy. Yeah.
Taking a quick break from our conversation to tell you about Applied Biosystems' QuantStudio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves in only 90 minutes. Digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. In fact, the microfluidic array plate technology enables consistent delivery of more than 20,000 microchambers. It's a great solution for anyone looking to quantify gene targets.
And Thermo Fisher Scientific has a suite of dPCR assays for applications like AAV viral titer quantification, liquid biopsy analysis, and wastewater surveillance. You can learn more at www.thermofisher.com/absoluteq or visit the Absolute Gene-ius webpage. Again, that's www.thermofisher.com/absoluteq or visit the Absolute Gene-ius webpage.
The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures. Let's get back to our conversation. Cassie, it's your turn. Cassie's Career Corner.
I need like my own little jingle for like Cassie's Career Corner, bum, bum, bum. But anyway, now we get to talk about all the cool, like fun, colorful things that have to do with your career. And I just realized now, like, we didn't really even get the chance to go into like even just like a 30-second overview. Like, what has your career journey looked like? Just so our listeners are aware.
It was actually pretty diverse. So I'm an international originally from India. And I did my undergraduate degree in engineering, interestingly, with a focus on industrial biotechnology. And then I came to the United States for my master's, where I started studying halophilic archaea. So these are salt-loving organisms that you see that turn hypersaline and membranes red in color. And then I decided to pursue studying halophilic archaea again for my PhD as well. It's been a long, drawn-out process because I've done 2 postdoctoral positions now. First one was in Boston College with Dr. Michelle Meyer, studying the oral microbiome and the non-coding RNAs in oral microbiome, and how the non-coding RNAs in certain pathogenic organisms affect pathogenicity. And then I moved to Stanford in 2019 to work with Dr. Sam Yang in the emergency medicine department. It's been a diverse set of projects to look at so far. So initially I started looking at how the host neutrophils responded to different stimuli, like bacterial, fungal, viral stimuli. And we were doing epigenetics to see, if we were able to get any epigenetic patterns for those. The lab has always been interested in potentially developing diagnostic tools for reducing the turnaround time in antibiotic susceptibility testing and stuff like that. When COVID happened, the focus switched to studying COVID, how it might cause extrapulmonary complications, severity, and even long COVID. And now we're doing other projects where going back to the focus of reducing the turnaround time for antibiotic susceptibility testing using microscopy. There's actually a different project where we used a digital PCR a little bit with Neisseria gonorrhea as well, but that's kind of the roadmap of how I got to this point now.
So you attended like your undergraduate studies, you completed those in India. You did your graduate work in the US. Did you not only make the jump from an engineering background to like pursuing like the genetics and things like that, but did you also make a total jump like to a brand new country as well all at once? So that's like a lot.
Actually, I'd never been out of my country before I came to the United States for the first time.
So what was that like? That must have been overwhelming.
It definitely was. I grew up in a city in India, and I did my master's and PhD at University of Connecticut, where it's in Storrs, which is a small town, college town. So there wasn't really much around where when I got there. So it was a pretty big change for me.
How did you navigate like some of the challenges that you probably faced when you were trying to make all of this adjustment at once?
So for one, at least when I was in India, in my undergraduate institution, we didn't have a lot of opportunities to do hands-on research at that point. It was just starting out. So a lot of the things that I learned theoretically, I'd never actually done until I came for my graduate program. Apart from that, just the way the whole exam structure itself was very different. So when I was in India, we were told that these were the classes I had to take, and we had a schedule that was given to me. I didn't really have the option to pick and choose classes at all. Just being able to pick classes, I was like, oh, I want to take this and that and that and this, and I want to, you know.
And that leads me to kind of a good question because a lot of our listeners, at least we're hoping, are a little bit either earlier in their career or they might even still be pursuing their studies themselves. Do you have any tips for like navigating like stress or like managing that? Because I mean, a PhD alone is a lot and it's a lot to work through. So what would you suggest?
I was very lucky. I had a good support system. It's essential. I was lucky enough that the gym at UConn was really good, and I had a good group of friends who would always want to go to the gym and play a sport or lift weights or whatever. So you are bound to get frustrated when you're in grad school. So you just need to find a way to distract yourself in a healthy manner.
Listening to kind of your overview of your career and everything like that, has your career gone the route that you would have expected? You've made some changes over the course of things. So is this ultimately when you first started like your studies all the way back in like undergrad, is this kind of the trajectory you had pictured for yourself or just totally different?
It's kind of on the same trajectory, I would say. One of the reasons I wanted to pursue a graduate career was to eventually become a professor. I was lucky to have really good instructors, both in undergraduate as well as in my graduate school, and my mentors all through so far. They've built me piece by piece to where I want to be, I think. So my current training with Sam has really helped me, just learn about the nitty-gritties of how a lab runs itself. Ever since I've been a staff scientist here, Sam has given me the opportunity to learn a lot of that.
And what is it about like professorship that you're so interested in? Is it the mentoring component? Is it like the research component? Is it a little bit of both?
It's a little bit of both, and I love to teach. I always knew I wanted to teach at some point, but when I was in grad school, I was a TA for most of my grad career, and I just thoroughly enjoyed, being able to interact with the students and finding new and interesting ways to keep them engaged and just, teach them the basics of what they're learning and all of that stuff. I think I love doing research, but I think teaching is what drew me into this.
Awesome. Well, this is like on a very minor scale for me, but like, so I tutor on the side. So, I mean, obviously I'm no professor, but like, I will say that, being a tutor, I'm typically working with students who are feeling very challenged with a subject or something like that. And then when you finally are able to connect with them on a level and explain something in a way that works for them, because everybody's mind works differently, it's just like, it's such a rewarding feeling, isn't
Yeah,
it?
I agree. I completely agree. And then towards the end of the course, when they come back and ask you questions, and you're like, that's interesting, you feel good about yourself, right? Just to be able to do that.
Yeah, it feels good to make other people feel
Yeah.
good. So at least I think.
Hey, you're shaping the
So,
next generation as well, too, right? The next people that come up in the field.
Yeah.
yeah, no pressure. So, what would be your biggest piece of advice for people who are either new to the field or even to yourself back when you were first starting out on some things, like even on your career journey? I know we talked about the importance of having like a good support system and all of that, but just people who are starting out in their career even.
I think I would say, you start with a particular interest, but I think you need to stay open to find the right path for you, at least in terms of the questions you want to answer long term, right? So, as I said, I started working with Haloarchaea and then the oral microbiome and now AST. Um, I feel like you need a diverse set of experiences to be able to really learn from all of that to, then be able to say, okay, this is exactly the question I want to answer in the future. So if you're able to get some of that from different places, definitely take it. Right? And in this day and age, I think interdisciplinary sciences are everything now. So it's definitely good to have that. Don't limit yourself to just bioinformatics or just molecular biology. If you can be both a bench and a wet lab scientist, go for it.
I love that. There's a lot of different skills that you can learn from a lot of different areas and apply in many different ways. So I think that's excellent advice. You refer to yourself as an analytical computational biologist, right? That ties into like bioinformatics. And I don't know a ton, admittedly, about bioinformatics myself. And maybe this is because I'm pretty detached from the field, but like I would assume that at times there are probably those who struggle to understand what the kind of the world of bioinformatics is, or kind of people who are maybe adjacent to that, but they're not totally familiar, trying to relate to it. So like, what is your method for explaining something of this nature to people who may not be totally familiar or as ingrained in it as you are?
That's a tough one.
You're like, I don't. You're like, I tell them to Google it and then I walk away.
That's a tough one. Bioinformatics is just a simple tool that one uses to understand the biology of things, right? All of the sequencing and all of that is just a tool to be able to decipher questions that we have not been able to answer before. Every day there's a new technology that's arising, and bioinformatics, I think, is just a way to make sense of what the technology is putting out. It started off with just DNA sequencing, understanding the genome, and it went to the transcriptomics and understanding the gene expression patterns. As new technologies arise, bioinformatics just works as a bridge between, the person who's generating the data and what the data is telling you.
So for those who might be interested in the field and learning more about the field, what is like a day in the life of an analytical computational biologist? What does that look like?
My current projects involve both looking at, sequencing data, genomics or metagenomics or transcriptomics, mass spec spectra for MALDI or LCMS. I'm trying to get some information from microscopy images. So it's like I feel like I'm providing the bioinformatics support for all of the projects in the lab, but I also get to be a part of every single project that happens in the lab, right? So you kind of have to prioritize which project needs to get done when, and then you're looking at different kinds of data. You're pretty much just staring at the screen all day, trying to write a line of code that, someday flows smoothly. Some days it's just like you have a barrier there that just doesn't let you code anymore.
So I would say it's two things. One, you have to have a lot of patience. And two, it sounds like every day is just a little bit different for you. Like you're involved in all these different projects. But I think that would probably keep things very interesting for you and very engaging at the same time.
Definitely. I'm glad that I don't have to do the same thing over and over. This way I get to learn a lot of new things as well.
Now we move to the part of Cassie's career corner. in which I ask you about what would be your best or proudest lab moment, do you think, so far in your career?
I feel like, science is a battle most of the time. Things don't always work out the way you expect it to work out on a daily basis. So every small victory, I think I'm proud of. I've had rough times, so when things work out, you just celebrate it.
Yeah, definitely. Okay, well then, since you've given me the perfect segue, thank you. So you said that things don't always work out. What would be your biggest lab oops or mistake? Or do you not want to own up to that on the podcast? And that's okay.
No, so so when I was doing my PhD, there was about 2.5 years into my PhD, there was some contamination in the lab that basically meant that I had to dump that project and start a whole new one. So that would be my biggest whoops in research. That got me to where I am today.
Yeah, all's well that ends well. Mistakes do happen, things like that. And then you mentioned like your support system and things like that, and like sports and all that. Has science influenced your life outside of your career or any hobbies that you might have? I do hear you like basketball, and I do hear that you speak like 10 million languages. Although, when you're not looking at numbers and the data and everything else, what do you enjoy doing?
I I like to, maybe it sounds a little bit like a cliche, but I when I can just focus on my son, I'm very happy just doing that.
Oh, that's great. Excellent. Anything you enjoy doing together? Or is he still like itty bitty
He's
still? So he doesn't.
he's not even 2 yet, so every day is different, and so right now that's outside of work. That's all I do now. So
That's awesome. Any plans on bringing science into his life at an early stage?
if he wants to, yeah.
Yeah.
I think
I
that's
love
the right
that. Well,
response for any
I
2-year-old,
think
right?
I think that's exactly right. Yeah.
That was Nikhil Ram Mohan, staff scientist at the Stanford University School of Medicine. Thank you so much for joining us for today's episode of Absolute Genius. Stay curious, and we'll see you next time.
Being an analytical computational biologist and working in bioinformatics makes you like a computational biological detective librarian profit wizard.
I like that. That's going on my Stanford profile now.
There it is.